about 8.5 years, and the museum itself just celebrated its 10th anniversary. Please wave at me if you have visited there before. Has anybody here visited the Reach? I know a couple people have. Good. I always ask that question when I'm, whether I'm presenting to children or adults, and I'm finding that a lot of people still are discovering that we're there. So our museum is located in Richland, Washington, right kind of central to all the Tri-Cities, and right next to the Columbia River. Our name when we first opened was Hanford Reach Interpretive Center, and I'll talk about why that is. But we, as I mentioned, have been open for 10 years, and so it's really great to come and visit and be a part of this program here. My experience, besides being at the museum for about 8 years or so, previous to that, worked mostly in outdoor education, and everywhere from Southern California to Texas to several places in the Midwest. And my favorite thing to learn about whenever I move to a place is about all the different interrelationships that we can find in the local ecosystems. And so my goal, wherever I go, is to engage in place-based education. I have to find the mouse here in order to advance. Perhaps I can do it that way. So what place-based education means to me is using our local community, using our local ecosystems in order to teach many of the standards-based curriculum that people need, that children need to learn. It's only in the past few hundred years that education has been kind of pulled out of the community and isolated in the classroom, as if you can learn about subjects without their context. And so I find that making those connections with people in the community, but also just your neighbors that are non-human, and getting to know the place is what really brings education to life. The picture that you're seeing, I'll have to tell you who took that photo, but first of all, if you've been at the Reach Museum, you'll remember that in the center of the building, in the main hall, you not only get a beautiful view of the Columbia River, but we also have a floor-to-ceiling, what looks like a map. It's a satellite view of the Columbia Basin Project. So all of the areas that are irrigated by the Grand Coulee Dam can get a really good view of the Hanford site and just all the surrounding areas. You can see evidence on that of the Ice Age floods. You can see the scars on the land. You can see old pathways that the river has taken millions of years ago. And every time I look at that satellite image, I learn something. And so I like to use it for student education, mainly as a reflection tool. I have students sit down and draw. What are the major things you see in that satellite view? Draw the course of the river. Note the different colors that you see. And then thinking about how that's actually a photo, that's not a map that's drawn, reflecting what would that have looked like if we could have taken a satellite view in 1805 when Lewis and Clark came through. What would still be there? What would not be there? And it's something, like I say, I learn something every time I do that, and new thoughts come to my mind. So after spending one morning, engaging in that with students, took my lunch break, went to my desk and pulled up social media like we do at lunchtime, and in my social media feed, this photo was there, and it was taken by an astronaut from the International Space Station. So it's astronaut Kayla Barron, and when she was up on the International Space Station, she said, "I've been waiting to get a photo of my hometown," and so here it is. So she snapped this photo out the window, and it just really touched my heart to see, someone who grew up in this area, made it out to space, and was still thinking about her hometown. I got the opportunity to meet her a couple of years ago too. She came back to Richland. We held an event that we invited teachers to come and have dinner with the astronaut and meet her, and she's a great, she really helped put things in context for me. So I have to credit Kayla Barron with this photo. What place-based education means for the Reach Museum is that it's really a part of our mission. The mission of the museum is to inspire learning by sharing the stories of the Mid-Columbia River region, its people and its contributions and impact on the world, which are profound and significant considering the work done at the Hanford site. And so that is woven into the story, but really the river is what winds through all of these stories and connects them. And it's something that we refer to no matter what stories we're telling. A lot of the programs that we do at the museum utilize the landscape itself. We have a trail that just goes through the museum grounds. We have about 17 acres of land just around the building, featuring a lot of native plants and a garden with an irrigation pivot and outdoor exhibits. The arches that you can see here are the center piece of a scale model of the solar system that extends throughout the Tri-Cities. So that represents the sun, and if the sun were that wide, about 40 feet in diameter, the orbit of all the planets are marked throughout the Tri-Cities, usually along the river trail. So that's an ongoing project that one of our partner organizations is still working on installing, but just a great example of how we use the landscape to teach. So we serve over 6,000 students each year with field trips and classroom programs, do a lot of outreach in the community, and then we hold a lot of events at the museum that are designed for families. Many of our exhibits are actually not all that family friendly or interactive for young learners, and so we make an effort to really do a lot of programs that allow for hands-on activities. And so the name Hanford Reach or Hanford Reach Interpretive Center refers to our job of interpreting the Hanford Reach National Monument, which is for about nearly 200,000 acres of land surrounding the Hanford site, including what we call the Hanford Reach of the river. That's that large bend in the river, but its uniqueness is in that it's the longest reach of the river without any dams. That has a big impact on the shape of the river, and of course for the life in the river as well, as well as the shape of the land around it. There are many threats to that river, to that reach of the river, and so the effort to turn it into a national monument, it was many, many decades in coming. We're going to be noting the 25th anniversary of the establishment of the National Monument next year. And some of the threats include there were efforts to put dams along this reach, to dredge it, to make it a shipping channel, but also just the agriculture that is located above. You can kind of see that image of the deer is behind it is White Bluffs, and uphill from those White Bluffs is a lot of agriculture, irrigated, that takes place, and that has resulted in some slumping of the landscape into the river, which really threatens the salmon habitat. There are many salmon reds in that area. And of course, you can see the image of the cocoon reactor on the Hanford Reach National Monument. There are 9 reactors. Many of them are cocooned, meaning they're just covered up in place, but still contain a lot of dangerous materials. So at last check, about 11,000 people go to work there every day to help make sure that, that none of that waste leaks into the river, and that is the primary focus of what happens now at Hanford. So all of these concepts, as you can imagine, in trying to impart the knowledge to the next generation is quite a challenge. These are very big issues. The river itself is huge. Its watershed covers nearly the same geographic area as the country of France, right? 250,000 miles in that watershed. And all of those things can affect the water, the quality of the water that we use here. So it's in order to promote a sense of belonging and a sense of ownership of something as large as the river, we take the approach of looking at one of the creatures that you can actually just pick up and hold in your hand, and that kind of brings it all to one point and helps it be much more accessible. So that is why we I've been really happy to be able to take part in the River Mile Crayfish study. And so explain today a little bit why crayfish, what makes them so perfect for this work, but also how this particular way of doing things really is a great way to bring some sense of agency to the students that are participating. They're able to contribute to real science and help solve problems that may seem small but have a big impact. The River Mile is a network of educators who engage in science to just learn more about water quality in the Columbia River watershed. This network got started back in 2008 at Lake Roosevelt National Recreation Area, and has been a project of the Lake Roosevelt Forum. It's been supported by grants from National Park Service, National Parks Foundation, and the Environmental Protection Agency most recently. And so many of the illustrations and teaching tools that you'll see were developed by the River Mile Network, but also by an agency called Engaging Every Student. And they created a complete curriculum guide suitable for grades 2 through 12 that is available for free on their website. I have one copy here if anyone wants to take a look while you're here. And like I say, that's freely available, and I'll give you some more information about that at the end too, in case you want to get involved with it. They also offer teacher training to be able to use the curriculum, and I say they, but we've been a part of it too at the Reach Museum. So now, as we go on to learn more about what makes these crustaceans so charismatic, I have to credit that I actually stole this title, Charismatic Crustaceans. I stole it totally from Dr. Alan Kolak at University of Idaho, who oversees the Crayfish Mercury Project, which I'll talk about more in a little bit. But the reason that they chose the crayfish as a study subject in order to look for mercury within the Columbia River watershed is just because of its charisma, right? It's kind of a creature that many people know, they have stories about, they have some awareness of, and if they don't have the awareness of it, they're usually pretty interested to learn more, right? A lot of times I show people these creatures from the river and they are taken aback. They're like, we have lobsters in the Columbia River? And so that's a perfect chance to engage in some conversation about what they are and how they fit into the ecosystem. But as Dr. Alan Kolak says, part of what makes them so charismatic is there's always the danger that they can grab you before you grab them, and that just kind of adds an element of fun to it. And they're also fairly easy to catch, but they make a great place, or a great subject for study in looking for mercury because of their role in the ecosystem. And the drawing that you see up there, I'll credit that to a Pasco High School student who, as we were engaging in some scientific illustration, kind of went off of the science a little bit and added their own interpretation. So let's meet this creature. There's over 300 species of crayfish, also known as crawfish or crawdads. Those are all just kind of nicknames for the same animal here. Over 300 species found in fresh water across North America. Worldwide, there's closer to between 500 and 600 species. And you'll find the most diversity of species in the southeastern United States. But here in Washington, we don't really have a lot of diversity. In fact, there's only one species that is native to the state of Washington. That's that one up there, the signal crayfish. And if you are looking at the posters that I've got available for you to take at the back of the room here, you'll see on the native side there are two others listed: the Snake River Pylos and the Pylos crayfish, and those are found a little bit more in the area of Idaho. They haven't been recorded here in Washington. But if they were, we may consider them a native species. It kind of is. There's a lot of scientific record that is lacking when it comes to knowledge about crayfish in the Pacific Northwest, and so that's part of why this study exists, right? To help fill in that information and fill out that data. And we look at their role in the freshwater food web. Here, this is, you can see their position kind of at the bottom of things. Their job as, here it says scavengers, but really they're opportunistic or generalist feeders, in that they will eat whatever falls to the bottom of the waterway that they're in. You can see they're designed for being on the bottom. They're not swimming to the surface, and so they find their food at the bottom, and so by nature, they're going to be scavengers. They'll eat a lot of detritus and what kind of just looks like mud because it's just waste from whatever plant or animal matter has come to the bottom. They take that and they turn it into food for many other creatures. If you spend any time around freshwater ecosystems, you're going to notice a lot of things going after and eating crayfish. You can find the evidence of it with the little bits they leave behind. So anything from fish and amphibians to mammals, including people. And then also a lot of birds will be like to feast on crayfish, so wading birds and diving ducks, but also gulls. And I notice around the Columbia River, I see a lot of gulls catching the crayfish. The first thing they do is they tear off those claws because I wouldn't want to swallow a creature that's got those big claws on them. And that leaves behind some ways for us to help identify at least what's out there, even if we don't catch them. So pictured here, just going to bring up the names here, including the goldeneye. There's a great picture of the goldeneye that dove down and found them. As we learn more about the crayfish, I find that the best way to learn about something is to sit down and draw it. Is anybody here into nature journaling or engaged in that at all? A little bit. The studies show that stopping to notice something contributes to your overall health because of mindfulness. But if you really want to learn about what you're seeing, taking a photo can be okay. It can help you remember, and you can use it as a reference later. But if you really want to learn about something, taking the time to notice enough details to draw it really increases the learning exponentially. So, to that end, I am going to offer, we've got paper and pencils here, and I encourage you to to engage in some scientific drawing, and I'll go over what that means here. So I have an assistant here who will help with passing those out if you need one, and if you'd like to engage in some drawing. There is no artistic ability required for this type of drawing, and you can even get your own specimen to look at. You are also welcome to draw what you see on the screen, but it's not quite as clear, and so I can bring around a few of these specimens. You can just kind of set them a space next to you. Or we can share. I have 6 in total, so we might be okay. If you're willing to come and share one. Just everybody else. Set it right there. They are. Okay. Is this one here? And anyone else who wants to join in, you're welcome to come. There's 3 more specimens right up here. And so let's talk about what does, oh, and I have to say, please don't touch them. The reason is they're very easy to break, and also they are preserved in alcohol, so not so great for your skin. What does it mean to draw like a scientist? I always remember A, B, C, D, E. So A is for accurate. Draw exactly what's in front of you, not from your imagination. Big, that means try to fill your whole paper, which may seem intimidating at first, but that will allow for lots of details, which is what D stands for, right? D is for details. The purpose of doing this is to notice all those details. So include as many as you can. Colorful. I did not bring colored pencils with me today, but you can label colors that you feel are significant. And then E is for explain. Explain with labels. That can also include questions that come up as you are examining this creature. Feel free to note any questions, and then it is great to use numbers as well as words when you are labeling. And there's different ways you can do that, right? You can use, you can just talk about the numbers of body parts you're seeing. Measuring can also be a way to do that, and I'm looking around because I'm realizing the rulers that I brought, I do have some ruler magnifiers, but they're on the back table along with those posters. Feel free to take one of those to use. And as you are sketching, I'm going to keep on going here, and I'm going to advance to the next slide in order to, we'll start talking about some of the anatomy and how that anatomy is used. And first of all, I do want to say too, if you take a look at this complicated looking creature and it's, and you're like, how do I even begin to draw? One little tip, which you'll hear a lot of art teachers, but also journalers use, is just to take a look at look for basic shapes. So if you're looking straight down at the body, you may see an oval. Go with those big basic shapes first, sketch them really lightly with light short strokes, and then you can always go over to refine your lines with darker strokes later. And that will help you just kind of get the big proportions. How long is the claw in comparison to the rest of the body, for instance? And so that can help you get started. And then, because you're making a nice big drawing, you can fill in those details too. So as we're talking about our basic shapes, that correlates to the major parts of the body. And like a lot of arthropods, the body is kind of divided into sections. And on a crayfish here, we're going to divide it. The back half, or towards the tail, is the abdomen, and the front half, which includes kind of the thorax and the head, we call the cephalothorax. It's the same thing that you would be talking about if you're looking at a spider too. They've got a cephalothorax and an abdomen. whereas insects kind of have more of 3 parts, head, thorax, and abdomen. You'll notice that, now I asked you not to touch it, but if you did, what do you think it would feel like? Would it be soft? Would it be hard? If you tapped it with your fingernail, right? It would be hard because it's covered with a shell, not a super strong shell, but a carapace, an exoskeleton. And exoskeletons don't grow as the animal grows. And so that means that they have to shed it periodically. As they grow, they molt. A crayfish that is less than a year old is growing really, really fast and will molt every few weeks to every few months. And then as they get over a year old, it's more like every once or twice a year. I have up here in the petri dish here, this is a molt from a crayfish a live specimen that I had. I took it when I noticed it had molted in the aquarium. I took this out, but then I learned later that that it's better to leave it in there because they will actually usually consume their own molt after they're done molting, and that just helps them reuse those nutrients to grow and refine their new exoskeleton. So we've got the carapace is what you call the sort of the shell over the cephalothorax, and we have it divided into 2 sections here on the image up here too. But then it does not include those claws, and we'll talk about why in a little bit. How many legs do you see on your specimen? I think almost everybody's got mostly intact. They might be missing a limb or 2, but how many legs on your crayfish?
I think I see 8.
It looks like 8, right? 8 of the walking legs. But then we do include the claws. The chelipeds are considered legs as well. So these are decapods. Decapod meaning 10-legged. And just like other crustaceans, pretty much all the crabs you find will be decapods as well. If we think about how they fit in, or how they relate to other crustaceans, you'll notice they've got that segmented abdomen, a lot like a shrimp, or like a roly-poly, which is also a crustacean. They've got that shell, and they are, as we mentioned before, they are living at the bottom of a waterway. So their major means of locomotion is what, would you say? How do they move around on the bottom? Using those legs, right? They're using those legs, including the claws, to climb and wedge themselves in between little rocks. They, because they're prey for so many different animals, they like to stay hidden. And you won't see them out much during the day or when it's light. So if you want to go looking for them during the day, the best way is to find rocks as big as your head that are out in the river, flip those over, and you're likely to find some. But they're going to stay hidden. And so, and many of them will be at a significant depth in the water too. With not a lot of sunlight going down there, even when they are active if they are active during the day, how do they find their way around in the dark? What kinds of sensory organs do you see, or something that you think might help them sense what's around them? Yes, I see people using the antennae, right? How many antennae do you see on your crayfish there? This is all part of the scientific drawing. Make sure you get your numbers accurate. And it might be hard to see on some of them because they're kind of stuck together, but 2 long antennae, and then the 2 pairs of shorter ones are called antennules. And those serve to help them taste and smell what's in the water around them. They also may be using those as well as some, if you see little hair-like structures on their legs, those will help them just sense pressure differences in the water, a lot like how fish sense what's around them. So they can, even without their eyes, They'll be able to sense what's going on around them and find their food, which is not that difficult since they eat everything. Their eyes, it may be hard to tell on your specimen, are on eye stalks, so they can protrude out from the carapace a little bit. They might be kind of pulled in though. And we'll flip them over in a minute here so you can see the ventral side and get a little bit more of the anatomy, including the mouth, because that's an interesting place to look too. If they are not walking, but they want to move really, really fast, what's their means of locomotion then? Yeah, swimming is going to be a lot faster, but they're not really designed for swimming. What they're going to do is they're going to pull that the abdomen quickly underneath them, sort of does the crayfish scoot like that, and that sends them zipping backwards in the water really fast. Typically will just wedge themselves between a couple rocks, put their claws out to show you they mean business. And so that segmented abdomen, you can really see how that strong muscle is going to be super important for keeping them safe. That is the part that people like to eat, right? That the abdomen is where the meat is. And the tail fan made of telsins just kind of helps with like swishing the water like a big fin. And how many claws do you see on your crayfish specimen? Okay, so two. There may be one or two that have lost a claw, right? Or there may be one that's got one really big claw and one teeny weeny claw. So what's going on there? Any guesses what happened? It's a regeneration. So as you can imagine, these get caught quite a bit. If a predator nabs them by the claw or by a leg, they can get away still, and they will regenerate and regrow that limb. It does take a long time, but for some of the, this one here, I think, or some of the ones that I've been able to keep in an aquarium, it's interesting to see after they molt, where there was nothing before, when they were just a missing claw, they've got one. After they molt, there's a little one starting to grow, so you can see it probably takes a long time. And let's take a look at the ventral side too. And the view up here too is showing what you can't see because their gills are underneath their carapace. So have you ever seen crayfish walking on land? They do that, right? They'll come out of the water if they need to change their location, especially if the water's starting to dry up, or to exploit different food sources. They may walk for a few minutes on land to go to a different waterway. As long as they have some moisture on their gills underneath the carapace, they can survive, but you don't want to keep them out for too long. And we call that little beak that comes to a point in between the eyes there. It's called a rostrum. Kind of serves as a protective function to keep those eyes safe. We've got a pointy part right there. And you can kind of see some little limbs in the front there that will, that called the maxillipeds and the maxillae. When on a live one, you'll notice that they are constantly swishing those. And I believe, my theory is that serves to push some water over the gills. If you like, I will come and flip it over for you if you're ready to see the kind of the uglier side, in my opinion. This is where you usually start getting the alien jokes. So you can look for where the mouth is, and it's going to have some jaws on the outside of its mouth, right? Called the mandibles. And I'm going to ask again, how many claws does your crayfish have? So take another look. You still think it's 2?
Yeah.
Yeah.
This one has 3.
Yeah, so if you look on some of the other walking legs, you might notice the ones in the front have tiny little claws too. And those are the ones that I've observed that they use mostly to bring food up to their mandibles. Their bigger claws, will work for catching something, but in order to actually pick up and bring the food to their mouth, they need to use those little ones. And it's fun to watch them eat because they just use as many legs as possible to bring that food up, and they look so greedy. If you're wondering if you have a male or female, I'll tell you where to look, right? You're going to look at where the abdomen meets the cephalothorax. So you'll see that most rear pair of walking legs. And then when you look at the underside of the abdomen, you'll see a lot of those tiny little appendages. Those are called swimmerets, although they don't actually seem to have much function when it comes to swimming. Those are there to, on a female, to hold the eggs. She will hold onto the eggs until they hatch. But males have them too. We don't really know exactly what else they do, what their function is. It may have something to do with steering as they swim. But if you see between the rearmost pair of legs, if you see an extra pair of swimmerets that are kind of enlarged, those are copulatory organs on a male, called the gonopods. If in the absence of those, you'll just see an opening, and that's where the eggs are laid. So, does anybody have a theory if yours is male or female?
I think we have a male.
You got a male? I think that's just a dark spot. Yeah. So those are that's a male. Yeah, what do you think? You got a male? Yeah, you do. How about yours? You got? Oh, okay, so you have a male there. That one is, yes, that one's a too. But we definitely find that more males go into the trap than females. A lot of people jump to conclusions about that. I don't think it has to do with intelligence or anything, but I think it has more to do with females being kind of more cautious and reclusive, especially during breeding season. They tend to lay their eggs in the winter, and so in early spring, they still might be, the females might be hiding out a little bit more, and not as likely to go into a trap. This drawing isn't super clear, but that just is showing the difference in the organs, and then an example of one that's got its eggs on it, and you can see how tiny that one is. So that might be the first year it's breeding. So when we are doing the crayfish study, what we typically do with students is we start with this activity, right? Just introducing what is the crayfish, how many of you have some experience with them, and where do they live, what's their role in the ecosystem, and getting to know their anatomy and adaptations a little bit more, because as when we actually go out and trap some, having that knowledge will help us be able to identify the species, male or female, and we like to take some measurements too of how long the body is. And so let's talk about which kind. Oh, and that's an example of one of the traps I typically use. Those are the ones I like the best. You can see there's a can in there, so a can of cat food is what we use as bait. But a lot of people use just any kind of meat, kind of whatever's in the back of your fridge that smells really strongly. Might actually bring them out, or some people even claim that you can just put bacon on a string and drop it in and they'll just grab right on, right? But we typically set out these traps overnight as they're more active at night time. And so in order to be able to tell what species you have, that's really the most important information as we're trying to learn what is the distribution of the native signal crayfish, and are there invasive species present in the ecosystem. The important thing to be able to identify them is to just take a really good look at the claws. And so if you look at the one that you have. Many of you might see, if you're looking at the edge of the claw, you might see some pretty prominent raised bumps all along the edge of those claws. Those are called tubercles, and they are going to be, if you see those bumps, then you're going to know that that is not a signal crayfish. The signal crayfish, which is pictured up on the slide here, has smooth claws, and the reason they're called the signal is because they've got that white or light bluish greenish spot right at the joint of the claw. As they get bigger and older, that gets more prominent. And from what I've observed, when you are looking down into the water and you see a dark shape down there, if it's a signal crayfish, those white spots will really stand out, kind of like headlights. And so I think that's why they're called signal crayfish. Their habitat that they like, cool water with a current, rocky bottom. And it's worth noting that they have never been observed being burrowers. They don't dig into the mud. And that can have a big impact on water quality. The other species that we have been finding in the Tri-Cities area, and unfortunately throughout the Pacific Northwest, is the northern virile crayfish. That's the one with the bumps on it, on the claws. It is not native to this area. It's native to northeastern United States and in Canada. And how do you think they got here? It's by people, right? Because they're not going to be walking across the Rocky Mountains to get here. So most of the problems with invasive species when it comes to crayfish is because people are transporting them to use as fishing bait, or a lot more often they'll be finding, or they'll be studying them in the classroom, right? If you order crayfish for science, you're not going to be getting, most likely, a species native to your area, and a lot of times people just don't want to euthanize those when they're done with them. Releasing them into a habitat where they're not native can cause a lot of problems. And so if you, looking at the habitat of the northern crayfish, they are known to burrow. They'll dig into the mud, especially when water levels start to go down, okay, which can be really variable in a lot of waterways, and that will just help them survive. If they can burrow into some moist mud to wait until the water level comes back up. If they're doing that, though, it can really introduce a lot of sediment into the water that wouldn't normally be there. So, I took this photo. This was significant just because during the pandemic, while the museum was shut down for a year, and we weren't really able to go and do programs at classrooms, I started learning more about crayfish and how to participate in the study. And so this was the first program that was going back to the schools to do, was introducing them to crayfish, getting them to do the scientific drawing, and getting ready to participate in the study. But beyond that, really the point is just to get them out of the classroom a little bit, interacting with their environment, and going to see how they can make a difference in their community. So engaging in some community science. The ideal program that we do gives a series of programs so that it really prepares the students so that when they go out into the field to collect crayfish, they are confident that they know what to do, and they know how to identify the species. So after visiting them in the classroom with some specimens that don't try to run away from you as you draw, I usually like to bring live ones too, so they can just get comfortable with how they move, what they look like. But then we have them come and visit the museum too. At the museum, we help to put the crayfish in the context of the freshwater ecosystem. We give each student an assignment to do a mini research study, to go out to the exhibits and learn about a particular freshwater plant or animal, and then we come back together and actually use string to make a visible connection among us all and create a big web to just sort of reinforce that idea of reliance on each other. And then we also do a population dynamics simulation in the form of a game called the Competing Crayfish game, and also instructions for that are in the curriculum. But that really kind of brings it to an immersive level where they are working as crayfish to collect food, and then you introduce an invasive species into the picture, and they're starting to really compete with each other. Sometimes it brings up a lot of feelings too, but that's all kind of part of it to help them understand why, how invasive species can be really damaging to the environment, and getting a sense actually in the numbers as they go through the simulation. And then finally, meeting them out at the river, collecting crayfish using the traps, and also just finding evidence of crayfish in the environment. As I pointed out, we do some measurements too, but really the most important is to find out what species are out there. We've been working a lot with a particular third grade classroom in Pasco, and but also this is really great for high school students because they kind of have a lot more of that background already, and they're more willing to reach in and grab a crayfish too, which is fun. One of the places where we've been collecting a lot of data, especially with the high school students, because it's a little bit of a distance, a little bit of a hike away from the museum, is Bateman Island. Bateman Island is significant. It's pictured in this image, which is also on one of the posters back here. It's right at the confluence of the Yakima and the Columbia River. And so on that delta, you've got this spot where we have a historic record of what was there in 1805 when Lewis and Clark came through. Clark, as they were camping near Sacagawea State Park, what is now that spot, Clark took a couple of men and kind of went scouting up the river to find the Yakima River, known as back then as the Tapteel, and they came across an island that we believe is Bateman Island, just a little ways away from the museum. They describe what they saw. They came through in October, I believe October 17th, 1805, and they recorded in their journals that the water was crystal clear and that there were so many salmon that they were just laying on the shore, right? And they were spawned out salmon. And many, many people were camped there, living in the tule reed huts, and picking up these fish and drying them out and smoking them on racks. And they give lots of details about what it was like there. But now, if you take a look at the temperature data that you can see on the poster there, because Bateman Island was used for cattle ranching, a causeway was built connecting it to the Richland side of the river and stopping the flow of the Yakima into the Columbia in that spot. So normally the temperatures are in the main channel about in the 60s during the summer, but they go up to 70s and 80s. You can see those red areas. Raising that temperature makes it really unsuitable for salmon and encourages species that like to prey on salmon. And so there is a movement among Yakima Nation fisheries, but also other entities that really want to breach that causeway. And so that's what the context is for the poster. This is Cooling the Yakima Delta. They're presenting their side of the case and why it would be great to remove that land bridge, restore the flow to create better salmon habitats. As that story is playing out, though, because it's going to take a little while before anything happens on that, we definitely want to take the opportunity to learn about other parts of the ecosystem too. And so we've been sampling crayfish, on either side of that causeway, just to try and see what data we can collect on that. And so far, pretty much all that we've been able to catch in those warmer waters are the invasive species. Love to use crayfish for outreach as well. But I wanted to take a look at the data map. So this is the entire watershed, and you can see every dot on that map is places where people have captured crayfish and reported them to the study. Typically students that are doing this. If we zoom in on the Tri-Cities area, and if I'm able to, I forgot to ask about this ahead of time, if we're able to link to the current ArcGIS map. Trying to open up a browser and do that. This data is publicly accessible, and it's a great interactive map to be able to tell what crayfish you can expect to find in your area. It's centered here on Lake Roosevelt, since that's where the study got started. And you can see a lot of collection has happened in Spokane and the Spokane area, around Portland, and then I'll zoom into the Kennewick area here, the Tri-Cities.
It's not changing on the
Oh, it's not changing up there?
screen.
So is there something I have to click on to bring up the browser?
Yeah, just so that you could,
Okay, yeah, sorry. Thank you for
is
your
this the
help.
browser?
Yeah, that's where we are. Thank you. So just real quickly, we can see the green ones are signal crayfish, and a lot more of what we're catching is the northern crayfish, the invasive species. So right here is where the museum is. We've been able to do a little bit of trapping here. This is Bateman Island, and then a lot of trapping right across the river at Chiawana Park, which is where we work with the 3rd grade class. So, but again, that is publicly available. One other place to note, and you'll see this on the poster map as well, high school students actually this isn't on the poster yet, but high school students in Lewiston discovered an invasion of red swamp crayfish right next to the Snake River. Actually not in the main channel, but in a pond next to the river. And so action is being taken to try and control that before they get into the Columbia River, or into the Snake and then the Columbia. Red swamp crayfish are native to Louisiana. So definitely not local, but what you are most likely to get if you order a science kit about crayfish. And then another significant invasion that has been discovered, and I don't think I can find it on here, but rusty crayfish have been noted in the John Day River for several decades, most likely related to a classroom pet that got released. So all it can take is just one creature sometimes to set off an invasion. So what I have learned as I've been taking part in this study, I feel like this has really helped me help students make connections. I love that they are going to their own neighborhood park to collect data. I love that they are asking lots of questions and trying to see how they can help with this. Spreading the word about how invasive species can be a problem and what not to do when you have a pet that you don't want anymore, right? Because that's how we start making a difference, is by helping young people teach their families. The Crayfish Mercury Project, I just wanted to give a shout out to them too. As I noted, they are looking for mercury within the Columbia River watershed by sampling crayfish. And so if you happen to know of a place where there are a lot of crayfish, if you're interested in participating or getting students to participate in this study, you may end up with a lot of the invasive species. Washington state fishing laws do not allow you to return live ones, if they're the invasive species, back into the water. So you got to do something with them, right? And if you're not going to take them home and eat them, then another option is to donate them to the Crayfish Mercury Project. If you've got a significant amount, you can stash them in the freezer, and they will send one of their researchers out to you to collect those, because that's a lot more data points that they can get. And we're finding concentrations of mercury in the water from a lot of different places in the watershed. So again, that's part of University of Idaho, their water studies department. And there's a flyer on the back table if you want to know more about that or to get involved. And also I'll just put a quick thank you to the sponsors who have helped us be able to present and continue this education program and offer it to schools regardless of their ability to pay for the programs. So that's come from Bechtel and from DTG, and also just ongoing support from the River Mile. And here's my contact information too, if you want to get in touch with me to learn more about how you can participate in the study, or if you just want to know more about crayfish and or the Reach Museum. So I think I have reached my time limit here, and we need to see if there's any questions. Yes.
What is the source of mercury, and are we seeing a trend that's going down?
So the source of mercury can be that is a good question. There are a lot of different sources. I know that mining has been one of the major sources. As far as an overall trend, it's really kind of localities. Just like any other fish, there are advisories about how many crayfish you should be eating, and so always check that if you want to know. Overall, what I've heard about crayfish or mercury concentration in crayfish, as long as you're not solely eating or relying on them for your major source of protein, and as long as you're eating the abdomen but not the head. I know a lot of times people like to slurp out that part too, but there's going to be a higher concentration of mercury in that part. The abdomen is a little bit better. So again, just keep an eye on those advisories to know if there's a concentration of mercury in an area where you're capturing them. And that information is available. The Mercury Project did publish a couple of years ago. I was able to find some data that they had readily available online, but you can always contact them for more information. And then, like I said, those fishing advisories can give you more information about where it's being found. Yeah, good question. Yeah.
In terms of just like the regenerative process and everything, is that only limited to like its appendages, or?
From what I understand, yeah, because like obviously if you cut one in half, it's not going to be able to survive. So I think it's really just the limbs, maybe even the antennae, but I haven't observed that myself yet. Yeah. I've been keeping, for several years, I've been keeping some of the signal crayfish, the native ones, in an aquarium to be able to have live ones to take out. So it's, a lot of times it's hard to keep them alive over the winter, but I have observed a lot of them go through the molting process. It can be pretty precarious for a lot of them too. Sometimes molting goes awry and they get stuck. And that's probably when a lot of them get captured by predators, so, yeah.
What is the average lifespan of a crayfish?
Yeah, good question, and I don't know if it's species specific, but, so I know that they grow a lot in their first year. I caught one in April that was this big, and it is now this big, so they can go really fast in their first year. And then, of course, slowing down a lot. I think the largest one that I have caught that, not including the claws, was about 12 centimeters from the rostrum to the end of the tail. And so I would estimate that could be up to like 7 or 8 years old. Just judging by how widely preyed upon they are, you won't find those very often, older than that is my guess. But it's one of those things that we don't have a lot of data on. So participating in this study and getting those measurements will really help us figure that out.
What do you feed them in the aquarium?
In the aquarium, I usually just rely on like algae tablets for bottom feeders, but I also give them some fresh stuff, sweet corn, carrots, and I give them little bits of fish if I have it, and then sometimes snails from the pond, throw in there, and they can eat those too. I gave the crayfish a couple pieces of pork tenderloin the other day, and they were so happy with them. They held onto them and walked around with them all day. So it was really adorable to see that. Yeah.
Have you noticed your crayfish have different personalities?
Definitely. There are some that are a lot more reclusive and some that are more aggressive, and that doesn't necessarily correlate to sex. And it doesn't correlate to how many claws they have, right? There's, I had a really aggressive one that was just a lefty, right? He had lost one. I think this was him over here. And but when he saw us come into the room, he'd come right out, and be ready to challenge. But others will just run and hide. So there's kind of two personality types that we've seen. And I did hear somebody say that that's true of lobsters as well. They've been observing lobster behavior and finding that there are kind of two classes of lobster behavior. So it'd be interesting to see what becomes of that study. Yeah.